I welcome you all, my dear students. Today in this video lecture, we will discuss about a very important topic on bacterial gene transfer. We have already discussed about mutation, about DNA damage in this course of MMB 201, and we have already also discussed about the types of mutation, types of DNA damage, and all the things. Now, in this lecture, we will discuss about how gene is transferred from one bacteria to another bacteria. Okay, so this is a very brief introduction about bacterial gene transfer. Gene transfer is defined simply as a technique to efficiently and stable introduce foreign genes into the genome of target cell. So it is understood. But because is the efficiently and stable introduce of foreign genes. Foreign genes means your gene of interest. That interest gene you want to work, some good gene that you want to work, and you want to insert in the next cell into the genome of target cell. Genome of target cell into the next cell, your target specimen. Okay. The directed desirable gene transfer from one organism to another and the subsequent stable integration and expression of foreign gene into the genome is referred as genetic transformation. So transformation means the once you have transferred the gene should be expressive in the target cell, then you will be called as this is a transformation. Okay, stable transformation occurs when DNA is integrated into the host genome and is inherited in subsequent generation. That means when in the stable transformation, suppose you are transforming one gene into a host cell, and you after transformation you cannot claim that this transformation is stable unless until you are observing that DNA means the gene is already inserted, that is inheritable in the subsequent generations or not. That means you have to study about 3 to 4 generations minimum to check whether the gene is successfully inherited or not in the generations. So then only you can call stable transformation. The transferred gene is known as the transgene. Transferred gene means the gene you're working with, means the important or good gene is called the transgene, and the organism that develop will successful gene transfer is called transgenic. Okay. Next, actually in this video, we'll talk in part wise because in bacterial gene transfer there are mainly three methods. First is the conjugation, second is the transformation, and last one is the transduction. So first we can start with conjugation. What is actually conjugation? Conjugation means conjugate, to sit one by one, means to make a pair, is called conjugation. Conjugate, it is the process of gene transfer via sex pili. So this is the most important things here. Pili. Pili means a bridge between cells of opposite mating types, means between two sex cell on male and female. If there is a bridge, is called the sex pili. Okay, so in the picture we can show it clearly. So gene transfer via sex pili between cells of opposite mating, means the male and female, are in physical contact, cell to cell contact of each other. So history means this is the scientist name, uh, Jusul Lederberg and Eduardatum is a very complicated name. Okay, so it's important for MCQ. They discovered first is 1946 the bacterial conjugation in the world of microbiology. So now the method. First of all, this is the stepwise method. one by one. You can have the picture here. First of all, this is the picture. Now you can see the methods in word. First thing is that recurs the presence of a special plasmid is called F plasmid. In the bacteria, there is a plasmid is very much important for conjugation. This plasmid is called A plasmid. When the plasmid is present, that is termed as A plus or a male cell, and when the A plasmid is not present, that will be termed as A minus or female. Is understood? The A plasmid is actually responsible for 25 genes. This is very important, that mostly code for production of sex pili. So that means if F plasmid is not present, so there is no question of conjugation. So if the conjugation need to be taken place in between the two bacterial cells, then the one cell should have the A plasmid, because A plasmid contains a number of 25 genes that mostly code for production of sex pili, and sex without sex pili you cannot success the bacterial conjugation system. A conjugation event occurs when the male cell extend his sex pili and attach to the female. Okay, this attached pili pilus is called pilus in Bengali, also we call pilus, is a temporary cytoplasmic bridge, is the connection between the two cell. Temporary cytoplasmic bridge, the cytoplasmic connection through which the replicating A plasmid, including 25 genes, is transferred from male to the female, because male is having the F plus plasmid and female is F minus. That's why through the sex pili or pilias or pilus, this temporary cytoplasmic bridge, through this bridge, A plasmid will be transferred from male to female. When transfer is complete, then the result is to male cells, because only male cells are having the A plasmid from the existing and the daughter and the another cells, the female cells. Once it is A plasmid is inserted, that will be termed as a male cell also. When A plasmid is integrated, when A plasmid is integrated within the bacterial chromosome, the cell is called an HFR cell, high frequency of recombination cell. This is very important. When A plasmid is integrated within the bacterial chromosome, that means A plasmid is the additional genome and bacteria is already contain one chromosome. When they fused or integrated, then the cell is called HFR cell, means high frequency. High H for high, FR means frequency of the recombination cell. High frequency cells is very much important for competent cell preparation. Competent cell is the another topic, we will discuss it later. So now in the picture you can see, this is the bacterial strain, this is the bacterial chromosome, okay, and this is the F plasmid. Okay, you can see this is the A plasmid, and this is called A plus cell, means the male, and this is the pilus, this is the sexually, because the A plasmid contain 25 number of genes, which is responsible for sex pili formation. So once sex pili is formed, pilius extract, sex pili is formed, you can see the bacterial in the from the male to female, the F plasmid is transferring to the temporary cytoplasmic bridge. Here is the temporary cytoplasmic bridge. This is the detailed step. Directly you can write it here. Then the A plasmid will be replicating and transferred here. Then both the cells will have, because this is already was the male cell, and once the A plasmid is transferred, this is also termed as the male. So two male cells having the another chromosome in the bacteria. So this is called transformation, conjugation. Okay. So now is the very important thing. This plasmid should be integrated within the bacterial chromosome. Here, you can see this is the A plasmid and this is the bacterial chromosome. Once this is conjugated, means integrated into the bacterial chromosome, the A plasmid, that this is the A factor, then the cell will be termed as the competent. High frequency is the high frequency HF or high frequency cell. That means this is the very important cell is termed as the competent cell for bacterial transformation. So this is the all about bacterial conjugation. I think you understood all the slide, and if it is not understood, you can watch repeatedly and you can ask me question. Okay, thank you.